Hello and welcome to the fourth in our series of Genome Technology Development webinars for this spring. I'm Mark Adams. I'm at the Jackson Laboratory and I'm the principal investigator on the Technology Development Coordinating Center, which is a grant from NHGRI to help coordinate the efforts of over 65 funded programs in the area of genome technology development, including nucleic acid sequencing and synthesis, single molecule protein sequencing, and genome technologies more broadly, things that we're going to hear a lot about today. The TDCC's goal is to help coordinate collaborations and enhance the impact of the GT programs, including helping get the word out on advances that are developed in the program labs. We also have a wide variety of other outreach activities that are available on our YouTube channel. And if you've missed the other 3 sessions that were part of this series, those videos should be posted by the end of the day today at YouTube at GenomeTDCC. Areas of direct RNA sequencing, single molecule protein sequencing, and we had a terrific talk on single molecule or single cell approaches on Tuesday. Today, we have 3 terrific speakers on the area of genome-wide regulatory analysis, which has really been transformed by new technologies that are now available. And you'll get to hear about that from our 3 speakers today. Please enter your questions in the Q&A box on Zoom. We will have a session for the speakers to respond to those questions after all 3 talks. So it's my pleasure to welcome our first speaker, Barak Cohen. He's the Alvin Goldfarb Distinguished Professor of Computational Biology in the Department of Genetics at Washington University in St. Louis. Barak, please help us off with your introduction to the field today.
Okay, thank you, Mark. Okay, so I'm gonna give just a brief introduction to the field. And since we're talking about gene regulation, regulatory genomics, this is gonna be a series of presentations mostly about non-coding DNA. So I thought I would start by giving you a sort of mental picture of how I think about non-coding DNA. This is supposed to be a cartoon of the DNA fiber snaking along down a chromosome. The DNA fiber is, as you know, mostly covered in histones so that it can be packed up and compressed into the nucleus. And all along the DNA fiber are these short regions of open chromatin, which are the interesting bits for what we're going to talk about today. For example, there are things like gene promoters down here, which are the sites of transcription that are going to transcribe the messenger RNAs that are going to actually make the open reading frames and construct the proteasome. These are modulated by things like enhancers, which can be nearby, they can be further away and loop in. There are silencers, which repress transcription. And these enhancers, one interesting property of them is that they can work over remarkably long distances. And in order to prevent crosstalk between gene regulation programs, there are elements like insulators, which are there to block the action of distally acting enhancers. So this cartoon is a reasonable mental model. It's a big oversimplification, but it does show 2 of the key things. The first is that when we're talking about non-coding RNA, or non-coding DNA, there's a lot of it. The open reading frames that are going to eventually make the proteome are only about 2% of the genome. So a whole lot of the rest of the genome is this non-coding stuff. And it's filled with these important pieces like enhancers, silencers, and insulators. Now, because there are so many of these cis-regulatory elements in the genome, that means that a lot of the important genetic variation in the human population is going to occur in these non-coding regions, simply because 1, that's where most of the DNA is, but 2, that's where a lot of the functional elements are. By some estimates, there's about twice as much DNA under selection outside of the open reading frames as there is inside the open reading frames. So there's important disease-causing heritable genetic variation that occurs out in the non-coding regions. And the going hypothesis is that they exert their effects by influencing enhancers, silencers, promoters, and insulators. So in that context, the important and major challenge is to be able to predict the effects of genetic variation. I say predict because I think the scenario that we're really all looking forward to is that one day all of us will have our genome sequenced, patients will walk into a doctor's office, and then based on the DNA sequence of individuals, we would like to be able to say not something non-trivial about those people. And if a lot of the important variation is going to happen out in non-coding, then we need robust computer models that can first identify where the cis-regulatory elements, so given an arbitrary DNA sequence, we want to be able to predict, is it or is it not an enhancer? And then more importantly, given a sequence that we think is a regulatory element and a genetic variant that we might observe in a person, we need robust computer algorithms that can predict whether or not this is going to make a change in expression. So predict the locations of enhancers and predict the effects of genetic variation that occurs within these enhancers. Okay, to do this, let's get just a brief picture of the anatomy of an enhancers. Enhancers are DNA sequences that are made up of constellations of binding sites for DNA binding proteins. So each one of these little boxes is supposed to represent a transcription factor binding site for a different sequence-specific binding protein. And it's really the collection of proteins bound to any given sequence that imparts its functions. So the trick with enhancers is to recognize when a collection of transcription factor binding sites is actually going to be a bona fide regulatory element and when it's just a spurious collection of transcription factor binding sites. So and then once recognizing that, we would like to be able to predict the effect of any arbitrary DNA sequence variant in these enhancers. Why is this hard? There are three challenges. The first is specificity. So here are a few numbers. The human genome is large. This is not really controversial. Transcription factor binding sites are short. This is also not controversial. But these two facts add up to a really big problem, which is that the genome contains many millions of occurrences of any particular transcription factor binding site that you might be interested in. And only about 1 in 1,000 of these are actually bound by transcription factors. So if here's a little model of the genome with millions of binding sites littered, most of these sites are going to be unbound, right? And only a small fraction of them are going to be bound. And worse than that, even fewer of the bound ones are actually going to have regulatory activity, right? So, and even worse than that, the same TF can often be involved in both enhancing and silencing. So the point is that context matters. All of these binding sites are identical, yet somehow the cell knows that only a small fraction of them should be bound and active. So where is that extra information coming from? There must be context outside of the DNA binding site that is important. One place where that context can reside is right in the DNA sequence of the enhancer itself. So let's say we know that this sequence is an enhancer that regulates this gene. It is not necessarily true that this sequence, which contains the same DNA binding sites, just arranged in a different order, orientation, and spacing, will necessarily be an enhancer. This is what we refer to as enhancer grammar. So there are an unknown set of rules by which motifs can be arranged in different orders, different orientations, different affinities, and different combinations. And only some of those combinations are active. So figuring out this grammar is a huge challenge in regulatory genomics. The second thing is that not just the local sequence, but the more global DNA context and the epigenetic context also matters. So if I take this enhancer and I put it down in several different places in the genome, here it might be active because it's in a region of open chromatin and it's looped in a way that it allows it to access its promoter. But here, maybe it's inactive because that same constellation of binding sites is now in a region of DNA methylation and it can no longer be active. Likewise, maybe here the enhancer resides in a region of heterochromatin where the histones are packed tightly and the enhancer becomes inaccessible partly because of the specific marks that the histones carry. Or the enhancer might be open and available, but it's being blocked by an insulator. So both internal matters of context, the actual arrangement of binding sites within enhancers, and the larger epigenetic context both matter. And figuring this out is going to be key to building models that can predict regulatory activity from DNA sequence. The third challenge is that there's what I call non-additivity at every level in this picture. So let's imagine a promoter that's being regulated by two different enhancers. If we delete this one enhancer, we will get an effect. Presumably the expression will go down. And if we delete this other enhancer, again, the expression might go down. But there's no reason to think that if we make the double deletion, that the effects of these two enhancer deletions will add up. In fact, most of the time they don't add up, and there are non-linear ways that enhancers combine with each other. Likewise, if we zoom in on an enhancer and come back to this picture of an enhancer as a collection of binding sites, if we mutate one binding site, we might get an effect. We mutate another binding site, again, we might get an effect. But there's, again, no reason to think that the double mutation is going to be additive in any way. And in fact, most of the time, because of cooperative interactions, competitive interactions, that the effects of different binding site mutations almost never add up. And again, if we zoom in even closer, if we look at just the binding site for this one transcription factor, we can well define the specificity of this particular protein. And we can know that if we mutate this C, we'll get a certain effect. And if we mutate this A, we'll get a certain effect. But again, the combination of mutations is unlikely or rarely additive. So you get non-additivity at the levels of DNA binding, non-additivity at the levels of interaction between transcription factors, and non-additivity between enhancers. So all of this adds up, pun intended, all of this adds up to a problem in predicting the effects of arbitrarily DNA variants that we might be interested in. Okay, so where is all of this going? The field has swung in a direction where we're putting a lot of hope into the big data approach. And that is largely because of advances in computing, particularly GPUs that have allowed powerful neural network approaches to artificial intelligence to sort of manifest in our field. And the idea is that we can take high throughput measurements of DNA sequence and the variants, of histone modifications, of chromatin accessibility, of chromatin conformation, DNA methylation, and many, many, many more, feed this into the neural network along with measurements of RNA expression and get predictions of regulatory activity. And there are some good examples in the literature of this, beginning to work, but it relies heavily on advances both in computing and on the artificial intelligence side, and in our ability to make these high throughput measurements. So I would say in terms of technology, there's 4 core enabling technology, the first being DNA sequencing. We all know that DNA sequencing is getting faster and cheaper pretty much by the minute. And that's enabled all of these seq technologies. I won't list them all, but there are many, many, many of these. And you're going to hear about 2 particularly cool ones as soon as I'm finished talking. DNA synthesis is also on another, not quite as steep, but also fast trajectory. Our ability to manufacture artificial pieces of DNA is getting better and better. And that allows us to make many, many targeted mutations in enhancers and their other types of regulatory elements. CRISPR, you all know about, of course, gene editing. This is going to be important for making targeted mutations within the genome in these enhancer and silencer elements. And of course, advances in computing, that as these data sets get larger and larger and more, that the computing needs to keep up. And that, again, the advent of GPUs and neural networks is largely where the field seems to be going these days. So with all of that, that means we now have these technologies that can measure where these regions of open chromatin are, who the transcription factors are that are bound, what the DNA modification state in terms of DNA methylation is, what the histone modification states, what the chromosome conformations are. And all of that is going to be fed in again to these neural networks. And again, we'll see if that's where the accurate predictions are going to come from. And I'll just finish by, to editorialize in one last point, because I can't help but editorialize, that the field has really swung, the pendulum has really swung sort of all the way to the left these days. So we're really talking a lot about machine learning, deep neural networks, and larger data sets. And the advantage of this type of model over more traditional sort of biophysical models where parameters that describe the binding affinities and cooperative interactions between transcription factors, the advantage really here is that the statistical power of these models is much, much higher. There's no doubt that if you have a large data set, much easier to fit this type of model and extract predictions. The downside of these types of models is that they are less interpretable. And here, the biophysical models have parameters that have very specific biological meaning. The parameters inside these neural networks, it's less clear what these, for example, there's no parameter inside here that would correspond to even the sequence of a binding site. That binding site would be broken up and distributed over many layers in the network. So these models become difficult to interpret. And so while I think that this is going to continue to be exciting as the new types of data come out, as computers get bigger, I do think there is still also some room for some hybrid approaches that can combine the interpretability of biophysical approaches with the more power of the statistical approaches. And with that, I will stop and turn it over to my colleagues. Thank you.
Thank you very much, Barak. That was a terrific and very clear introduction. Our next speaker is Bryan Venters. Bryan is at EpiCypher. EpiCypher is one of several companies that have received small business research grants from NHGRI that are part of the Genome Technology Development Program. Bryan is the director of genome technologies there, and prior to joining EpiCypher was an assistant professor at Vanderbilt University. Bryan, over to you.
Okay, thank you. All right, well, thank you everyone online for joining us today, and thanks to the organizers at the NHGRI for putting this webinar on. So today I'm going to tell you about two recently developed technologies, CUT&RUN, CUT&Tag, for epigenome mapping. So epigenomic mapping, of course, is important for understanding cancer research. And the reason why is because as a whole, the epigenome is often dysregulated in disease and cancer. So what are these epigenetic features? So if we zoom in on the chromosome and decompact it into the beads on a string, the two main features that are typically studied are nucleosomes, their positions, and histone modifications, as well as DNA methylation. And as you can imagine, because the epigenome is important for the cell identity and maintenance of over 200 different cell types in the human body, they're subject to immense regulation with readers, writers, erasers, and remodelers, which slide nucleosomes around. And there's an immense amount of clinical efforts in developing drugs to combat dysregulated epigenome. So how do we actually measure these features? Well, historically, a technology called chromatin immunoprecipitation that was first described 30 years ago, which is now used in more recently since around 2006, coupled to high throughput sequencing, such as Illumina or other platforms. And the way this works is you cross-link the cells, then you prepare a lysate, and then you isolate the chromatin, and then you add an antibody to fish out the target of interest out of all that chromatin soup, and you prepare a library and sequence it. However, in large part because of the sample preparation limitations and cross-linking and other factors, the data quality remains relatively poor. The throughput is low in large part because sonication is not particularly easy. It requires a lot of optimization. And because of the noise, you have to sequence much deeper than you would otherwise. Recently, in 2017, CUT&RUN was described by Steve Henikoff from the Fred Hutchinson Cancer Center, which is built on a immunotethering technology that was described by Ulrich Lemley. And the way this works is, first off, it's much different than ChIP-seq because you're not lysing the cells. It's an in situ assay where you immobilize intact cells or intact nuclei on magnetic beads, and then you permeabilize the cells, and then you add in the antibody against the target that you're interested in, and then you add PAG eminase, which the protein A, protein G moiety binds to the heavy chain of the antibody, and then the eminase is activated by adding in calcium, and then it very cleanly, kind of like a molecular sieve, cuts, and the cut fragments elute out of the nucleus. And so then you prepare a library from these, the DNA fragments that you recovered. And CUT&Tag is not much different in the workflow. However, the key difference is the enzyme is not a, instead of eminase, it's TN5, which you may be familiar with because it's used in chromatin accessibility profiling in assays like ATAC-seq. And TN5 is a transposase that has cut and paste activities. So it's cutting, but also adding a DNA fragment to the genomic segment. And the reason why these technologies are exciting is because they're orders of magnitude improvements over key specifications, such as the amount of material required. 10 times less materials required for CUT&RUN and CUT&Tag. And because the signal is significantly better because of this molecular sieve effect, you don't have to sequence as deeply. So you can get away with an order of magnitude less sequencing. So that means less expensive experiments. And at EpiCypher, we developed spike-ins, nucleosome spike-ins early and E. coli spike-in late, and I'll talk about the nucleosome spike-ins in a moment. But with these two technologies, how do you know which one to choose? Well, there's really two key questions you want to ask. First is what's your target? And if your target is a histone modification, then you have the option between CUT&RUN or CUT&Tag. And so then you ask the question, well, how many cells do I have to work with? If you're working with primary tissue, you may not have as much material, or clinical samples, you may not have as much material. And so CUT&Tag may be the assay of choice because it does well at very low cell numbers. But if you have plenty of material to work with, for example, from a cell line, then CUT&RUN is the generally recommended approach because it's more broadly capable because it can profile both chromatin-associated proteins such as transcription factors or chromatin remodelers, whereas CUT&Tag really doesn't do well with those targets. And contrasting CUT&Tag once again with CUT&RUN, it's a bit more of an advanced workflow. The handling is a bit different, but it's more streamlined because you don't have to do library prep. Okay, so we've talked about some of these features. And just one thing I want to mention, I'll circle back to this at the end of the talk, is what's really exciting about CUT&RUN and CUT&Tag as a platform is just in the 4 to 6 years that these technologies has been out, we've seen an extension of the capabilities of this tech. And however, it's all, whenever you're developing tech and talking about technologies, there's always a limitation. And so we want to discuss those. And like Chip, CUT&RUN and CUT&Tag are limited by the reagents, the key reagents, such as the antibody. And two key things you look for when you order an antibody are specificity and efficiency. So efficiency is just how tightly it binds to its target. So at EpiCypher, we produce a ton of nucleosomes that are semi-synthetic nucleosomes for a variety of applications, such as chromatin remodeling, FRET assays, and things like that. But the nucleosome that I'm going to focus on today is a nucleosome that contains a barcoded DNA segment, and that allows us to deconvolute its identity after sequencing. And so we can make these nucleosomes with defined modifications. And I just want to mention that this work has been really generously supported by the NIH. Okay, so we undertook a broad survey, and this was all CHIP and Luminex-based experiments. And to make a long story short, it's a tragic story, but 70% of commercially available histone modification antibodies are not fit for purpose because they fail just a simple criteria of specificity. And so the two criteria we used, or recovery. So in order for an antibody to pass this survey, it had to have less than 20% cross-reactivity, meaning that it found its advertised target, such as K4 trimethylation, 80% of the time. And it recovered greater than 5% of the material that you put in. And so we tested hundreds of lysine methylation and lysine acetylation antibodies. As I said, most of them failed. And all this work has been published, and there's a website, chromatinantibodies.com, that you can go to if you want to find a high quality antibody for your target of interest. Okay, so I talked about these nucleosomes, these semi-synthetic nucleosomes, and this is just an example of a panel of 16 nucleosomes that each have a methylation at one of these locations, lysine 4, 9, 27, 36, or 20. And the reason why we spike these in is threefold. They test the antibody performance in the assay, and it's really important to test these antibodies in situ, in parallel with the assay that you're performing, because they can behave differently depending on how you're measuring them. Of course, the information you get out of it can be used for normalization, and also just tell you whether the assay worked or not. So what does this data look like? So we took the best-in-class antibodies from this study, and what I'm showing you here is a heat map readout of the specificity. On the left is CUT&RUN, on the right is CUT&Tag. Each column is an experiment. And so, for example, in CUT&Tag, the first column is our negative control IgG. We don't expect it to enrich for any particular antibody. But if you take H3K4 trimethylation here, the on target is set to 100%. You can see it's very specific, but there are some differences. And for most, the good news is for most targets, there is a good commercial antibody available. And so as controls, we always look at IgG as a negative control, because one of the limitations of CUT&RUN and CUT&Tag is there's a modest accessibility signature that you can see here. K4 tri is a positive control for regions of the genome that tend to be transcriptionally active. And on the other hand, K27 tri methylation is a histone modification associated with transcription inactivity. And so they're mutually exclusive, and they show the expected pattern at promoters. But what does it look like at a specific location? So if we show all 15 of these marks with the negative control, and then I've got K27 acetyled down here, what you'll notice is that the methylation states are strikingly distinct. And Barak talked about enhancers. You can see that two histone modifications linked to enhancer signatures are H3K4 monomethylation, this peak here, and K27, but not H3K4 trimethylation. These peaks here for K4 trimethylation are associated with promoters. Also, you see the K36 monodiet tri series, that they're genomically distinct. And even more striking, the K27 series, that's all written by a single complex, the PRC2 complex, but they're kept genomically distinct by the cell. And so then we asked the question, we were excited about the telomere-to-telomere consortium publications that came out recently. And so we wanted to ask the question, do these technologies have access to highly heterochromatic regions of the genome, such as paracentromeres and centromeres? So again, I'm showing you all 15 of those data tracks and for CUT&Tag, and we mapped them to the T2T build, although I have mapped with an asterisk because this is short read sequencing. And although we can map the reads to a specific chromosome centromere, within the centromere, it's highly repetitive. And so the substructures we really don't know those. So that's a caveat to this. But strikingly, the expected mark that is known to be enriched at centromeres is H3K9 dimethyl right here. So you see that, and you can see that maybe there's some other marks enriched there too. And there's a load of activity in the adjacent pericentromeres. And I just want to point out the builds that are most frequently used right now, HD19 and HD38, a lot of this information is completely missed. Okay, so we collaborated with Ari Melnick from Weill Cornell to understand B-cell lymphoma and the epigenomic reprogramming underlying that disease. And so we performed CUT&RUN on these marks that I have shown here. And again, you can see just the striking patterns of K9, the K9 series, K9 tri and K9 di. They form these broad domains of signal enrichment, and they're interspersed with other marks such as K27 trimethylation. And another application of CUT&RUN is in drug treatment and drug stimulation. So this is the classic breast cancer model, MTF7. 500,000 cells were used in this experiment. And we have negative control antibody IgG and another control antibody here. And we use 3 antibodies for the purposes of sort of rigor, reproducibility, and validation. And you can see that upon stimulation with estradiol, they give essentially the same genomic pattern at roughly 10,000 peaks. And to further validate the data, we looked for the corresponding motif for this complex. And you can see that there's really strong correspondence of the ERE motif with the CUT&RUN data. And then if you compare 2 different antibodies against the N-terminus or the C-terminus, there's good correspondence there, as well as SRC3, which is a coactivator of hormone response proteins. So the question remains, what if there's no antibody for the target you're interested in? Or even more complicated, what if you're interested in sort of histone crosstalk or multifactorial recognition? And so just on the histone tail, it's decorated by a variety of histone modifications. And there's proteins such as BPTF that have domains that recognize, say, a methyl group and an adjacent domain that recognizes an acetyl group. So how does this work? How can we study that? So BPTF is a part of a complex, a chromatin remodeling complex called NERF. And so you can see BPTF in the two domains here. And so what we did in collaboration with Catherine Musselman from the University of Colorado was we generated a construct that contained a GST tag and the two domains that recognize K4 trimethylation and then acetylation. And we synthesized nucleosomes that this protein is known to interact with. And then we did a cut-and-run experiment and asked which one does it prefer. And this is the heat map. And just jumping to the right here, you can see that the nucleosome that it prefers is the one that has both H3K4 trimethylation and acetylation. By far, it prefers that one. But what about other nucleosomes? So we then performed an in vitro assay called DiPhil, and it's high throughput. We tested 65 nucleosomes. And again, you see the nucleosome by far that it prefers is the combo mark. So what does this look like in vivo? What does the chromatin patterns look like? This is a genome browser view of the antibodies against the targets of interest, the histone modifications, endogenous BPTF. But then when we add the tandem reader in, you can see that it largely recapitulates the endogenous BPTF locations. But what's really interesting are the exceptions that I've shown here in red. You can see that there's both prerequisites for BPTF, but endogenously, it seems to be negatively regulated. So that provides insights into the biology. But the exogenous finds it because it has those two prerequisites. And on the other hand, when you have just acetylation, it doesn't seem to care, either endogenously or exogenously. So just to wrap up, I talked about the features, advantages of cut-and-run and cut-and-tag, and how they can be applied to clinical research. And there's a variety of cell formats that are compatible with this. And so if you want to get started on this, I have some links here. And just looking forward, just in the last 30 seconds, what's really exciting is, again, in a short period of time, just in the last 5 years, you've seen, since these technologies were reported, an extension of these capabilities in cut-and-run, for example, single-cell cut-and-run, automation, and in cut-and-tag, spatial epigenomics is really fantastic. So imagine an intact tissue slice, and then you can map on that slice, retaining spatial information where a particular histone modification is. And looking forward, as far as new frontiers, working with, I mentioned clinical samples. So FFPE material is a routine way to preserve clinical samples, clinical material. And we're seeing advances on applying cut-and-run and cut-and-tag like technologies to FFP material. And of course, multi-omics and DNA methylation, combining the two. And you'll hear Nick talk more about DNA methylation as well as long-range sequencing. And so with that, thank you for your attention, and I'll hand it over to Mark.
Thanks a lot, Bryan. Appreciate that importance of controls and knowing what your reagents are. Really key points in all of this. I'll remind you, folks who are participants, if you have questions for any of the speakers, please put them in the Q&A. Our next speaker is Nick Altemose. He's the Howard Hughes Medical Institute Hannah Gray postdoctoral fellow at UC Berkeley. But within about a month, he's going to be starting his own lab in the Department of Genetics at Stanford and as part of the Chan Zuckerberg BioHub. Nick, welcome. Looking forward to hearing your talk.
So I'm really excited to talk to you today about this method that I helped to develop called DiMeLo-seq, which allows us to map protein DNA interactions and CpG methylation on the same single chromatin fibers using long-read sequencing technologies. And what really motivated the development of this technology was a sort of burning biological question that I had. Which was, where do centromere proteins localize within these large repetitive domains that house the centromere? And these domains at every endogenous human centromere are made up of these 171 base pair repeats that go on for between 300 kb and 5 megabases on different chromosomes. But we know from sort of early imaging studies that the centromere proper, the place where the kinetochore assembles and attaches each sister chromatid to the mitotic spindle, the centromere is only occupying a subset of those repeats within any given centromere region. So what we really wanted to explore was which subset is it, and why are those centromere proteins localizing there and not anywhere else? And also, what does that have to do with other epigenetic marks that we might be able to measure? So the reason that this became a burning question is because we've recently assembled across all of these super repetitive centromeric regions for the first time. As part of the telomere to telomere consortium, I helped lead the team that went and annotated all of the centromeric and pericentromeric repeat sequences in this newly truly complete reference sequence. In total, it adds up to about 190 million base pairs in the genome, which is about 6.2%, and for reference is about the length of the entire chromosome 4. So there's quite a lot of new material that's been added to the reference sequence. Most of it is made up of these super highly repeated sequences. There's a lot of really interesting biology happening in these regions. I mean, we all know centromeres are really critical for avoiding cancer, avoiding chromosomal abnormalities, and avoiding cell senescence. So in order to do this biology and understand how these regions are regulated, we need new tools to be able to do that. So the challenge that Bryan alluded to was that when you try to use short read sequencing technologies to map protein DNA interactions in highly repetitive regions, you run into some issues. So one thing that you can do is you can map a short read to a repetitive region if you happen to overlap a mutation that distinguishes one repeat copy from its neighbors. But if you're not so lucky, there are these large regions between markers that we call marker deserts, where short reads can't map uniquely. They will find equally optimal mapping locations across these entire regions. And so the best we can sort of do with short reads is either we average across all of these marker deserts and get sort of a very low resolution view of what's happening in these regions, just an average view, which can span megabases. Or we can be very, very stringent and only look at those reads that overlap markers, and then we get a very sparse view of what the protein DNA interactions would look like in this region. So what we were motivated to develop was a long read sequencing approach that could bridge across these marker deserts and teach us what was going on in these regions. So the tool that's really made this possible, in our case, was Oxford nanopore sequencing. And it's this really revolutionary method for sequencing DNA, which happens on a flow cell, which is about the size of a candy bar. And this flow cell contains thousands of tiny microwells, which contain protein nanopores, which are embedded in a membrane. Now, the way that sequencing works is a motor protein will unwind DNA and feed it a single strand of it through this protein nanopore, while a voltage is applied across this membrane, causing ions to flow through the nanopore. As that single-stranded DNA passes through, different bases impede that ion flow in different ways. And so the raw signal that you're getting out from a nanopore sequencer is current over time. And the way that each base affects the current is distinct, and using some very clever machine learning approaches, they're able to decode this electrical signal into a sequence of DNA bases. And what's more, they can not only tell the 4 bases apart from each other, but they can tell when these bases are modified, because modified bases will impede that current flow differently, and they have a signature that you can detect. So you not only get the sequence of your DNA, you get this meta layer of information that tells you where all the base modifications are, like CpG methylation. So we looked to existing technologies for mapping protein DNA interactions, and saw that there was a great need for something like the method that we developed. So in short read world, which I'm showing in the left column, if you're interested in just any protein DNA interaction, you just want to know where things are touching the DNA, but you don't really care about the identity of those proteins, you might use a method like a TAG-seq, where you're bombarding the chromatin with TN5 transposase, which I illustrate here as little molecular scissors. And those are only going to cut the DNA at accessible regions where a protein isn't already bound. And so by looking at the ends of these fragments that you sequence with a short read sequencer, like an Illumina sequencer, you can determine where the accessible regions of the genome were. However, if you're interested in a particular protein and you want to know where that is bound in the genome, you can use a method like CUT&RUN, which was introduced beautifully in the previous talk. And in this method, you're tethering these molecular scissors to an antibody which binds specifically, hopefully specifically, to your target protein. And what that does is it releases fragments that you can then recover, amplify, and sequence on your short read sequencer. In long read world, we absolutely don't want to be adding molecular scissors to our DNA and fragmenting it into little bits, or we wouldn't have long reads. So an alternative approach is to take advantage of the fact that we can measure these DNA modifications. And instead of bombarding the chromatin with molecular scissors, we can bombard them with methyltransferases. And I illustrate these as little molecular permanent markers. These can add methyl groups either to cytosines or adenines. And these methyl marks are very permanent. They're very stable, and they can be read out very clearly by the nanopore sequencer. And importantly, these methyl marks will only be deposited if there's not already a protein bound to that site. So the long read equivalent of ATAC-seq is FiberSeq, or there are other related methods that use similar approaches like NanoGnome. But with FiberSeq, they use an adenine methyltransferase, and they mark all of the accessible DNA with that enzyme. And the reason to use an adenine methyltransferase is because eukaryotic genomes typically don't methylate their adenines. We methylate our cytosines. And so if you add this mark to the genome and read it out, you can know that if you see a methyladenine, that was exogenously introduced, and that means it was marked by this enzyme, and it didn't come from the endogenous DNA methylation. So what we wanted to develop was a method for tethering these little molecular permanent markers to an antibody bound to the target protein, and only marking around where that protein is bound. And then we'll use a nanopore sequencer to read out that modification in order to infer where the protein binding event was occurring. So we developed this method, DiMeLo-seq, which stands for directed methylation with long read sequencing. And this was work that was done in my PhD lab with Aaron Streets in collaboration with Aaron Straight at Stanford and the other wonderful trainees on the paper, Kaushik, Owen, Rachel, and Annie. And the basic protocol is a bit similar to CUT&RUN. We permeabilize nuclei, we then flow in a primary antibody that binds to our target protein. Then we tether using protein A, our methyltransferase enzyme, this adenine methyltransferase. We do wash steps to get rid of all the unbound stuff. And then one of the tricks is that we add a methyl donor group called SAM for S-adenosylmethionine at the very last step after all the unbound stuff has been washed away, and that activates the methylation. So we deposit these methyl marks around the protein's binding footprint. And once that's done, we just extract the DNA by normal high molecular weight extraction methods. We sequence it by normal library prep and sequencing methods on a nanopore sequencer. And then we read out that modification information, and we simultaneously get methyladenines and methylcytosines. And from that information, we can infer the binding events, and we can map these reads even to the most repetitive regions. And what I'll say is this was a simple enough idea, tether a methyltransferase to an antibody, but it took an enormous amount of optimization. We had to test over 100 different reaction conditions to get from some very unsuccessful initial trials to a final, robust, very optimized protocol. And what's really cool about this method is that on the same single molecules of DNA, we can jointly measure where endogenous CpG methylation is and where protein DNA interactions were. This is really important for a lot of DNA binding proteins for which DNA methylation can abolish their binding. And one such protein is CTCF, which is a zinc finger protein that binds a very specific and well-characterized motif in the genome. But when that motif gets methylation marks on its cytosines, that will abolish or greatly reduce CTCF binding. And if we look at our DiMeLo-seq signal in aggregate at CTCF sites, we can see that's the case. Wherever we're seeing a spike in methyladenine, indicating that CTCF is bound, we see a strong dip in methylcytosine, consistent with what we know about how this protein binds. And what this signal also shows us, because we know exactly where the CTCF binding motif is, it shows us sort of the reach of the methyltransferase enzyme around this binding site. We can model this decay of methylation with distance as an exponential distribution with a half-life of about 170 base pairs. That gives you an idea of the resolution that's achievable with this method. And what I think shows off a lot of the advantages of this method is a non-centromeric application. Here again, we're mapping CTCF, but we're using the DNA sequence information from these long reads to phase them into haplotypes. So we can tell, did this read come from the chromosome that was inherited from the XX parent or the chromosome that was inherited from the XY parent? And looking across these plotted here, each gray line is a read, each red dot is a methyl CpG that was detected, and each blue dot is a methyl A that was detected, indicating that CTCF is bound. This is zoomed into about a 5 kb imprinting control region on chromosome 11, where we know that the haplotype inherited from the XY parent tends to be heavily methylated and not get bound by CTCF. And the opposite is true for the other haplotype, and that's what we see in the data. And what's really cool is that we also have reads that span across multiple CTCF sites, which can show us how they're jointly occupied on the same DNA fibers, which is something that bulk methods, especially short read methods, can really struggle to do. So as an example, going back to the original motivation of mapping centromere proteins in these highly repetitive regions, here we're looking at the Y chromosome centromeric array. It's the smallest in the genome. It's about 320 kb, and it's sort of annotated here in this red block. That's where all the repeats are. We can infer where CpG methylation is in this track, and we see that there are a couple of dips. And if you do cut and run with an anti-SEMP-A antibody, you either have that unfiltered averaging approach, which is shown in gray, where we get just kind of an overall signal showing, yes, SEMP-A is enriched somewhere in this array. Or you can do the really stringent filtering thing that I described, that's shown in black, but then your signal becomes very sparse. And it looks here like there's an association between where SEMP-A is localized and this area of low CpG methylation. But it's hard to definitively say with that sparse data set. And so now, in collaboration with Professor Karen Miga and her student Dan, we've been generating nanopore ultra-long reads, which can span hundreds of kilobases. We now have reads that actually go across the entire Y chromosome centromere array. And looking at the methyladenine signal, which is shown in this upper track in green, and simultaneously the methylcytosine signal, which I've split into this lower track in red, and all the reads are in the same corresponding positions. What we can see is that there's a very tight correspondence between where SEMP-A is localized and where this region of hypomethylation is. And we can start to see exactly where these centromere proteins are, and we can count their density. We can see, what fraction of nucleosomes contain this histone variant SEMP-A and specify where the centromere should be. And what's more, we can zoom all the way in, and we can start to examine exactly where these nucleosomes are and model that. So here, zooming into individual reads, each green line is a methyladenine, and each blue rectangle is where we might infer that there's a centromere protein A containing nucleosome. So while we developed this method in order to map these interactions in repetitive regions, it's actually a pretty versatile tool for studying chromatin biology and genome regulation. And it allows us to map multiple interactions on the same single DNA molecules. Because we don't amplify the DNA, we can infer interaction frequencies at each site across cells and jointly measure endogenous methyl CpG. We can map haplotype-specific interactions. So I did want to talk about some of the limitations namely that we require a lot of starting material. Unlike the short read methods, we can't amplify this DNA. We're having to sequence native DNA in order to read out these methylation marks, which can't be amplified yet. So what that means is you need to start with a lot of cells. And right now, we recommend starting with at least about 1 million cells, human cells per replicate. And the other thing is we're not only sequencing the regions that are bound by the protein, we're sequencing the whole genome every time. And so for large genomes, that resequencing of a whole genome can get quite expensive. So there are ways that you can target specific regions like adaptive sampling with nanopore, and we're starting to implement those as well. We're continuing to extend the technology to reduce those input requirements and improve things like single molecule sensitivity. We're trying to combine it with other measurements like DNA accessibility on the same molecules. And of course, we're trying to map multiple proteins simultaneously on the same chromatin fibers. So these regions of the genome that I discussed are sort of the new frontiers. We want to catch up to the rest of the genome in studying how these are regulated. And for that, we have to continue developing new tools. And that's what my lab will continue to work on at Stanford University. And I'd love to talk to anybody who's interested in working with me. Thank you very much. And I look forward to any questions.
Wonderful. Thank you, Nick. Folks, please enter questions in the Q&A so I don't ask all of them. That's the consequence of you're not asking. Let me start, Nick, with you. You talk about needing 1 million cells. That's 2 million strands of chromosomal DNA from 1 million cells. How many actually get read out, and how much information do you get sort of per strand, and how do you average information across strands, and what happens to all the other ones that you don't sequence?
Yeah, well, it depends on how much money you want to spend. You can sequence a large number of them if you'd like, but really, in most applications, you're probably sequencing 1 in 10,000 molecules at best. And part of the reason for this is that the rate limiting step on the sequencing flow cell is getting the motor protein to attach to the nanopore and start translocating the DNA. And, you know, one way of improving your pore occupancy and making sure you're getting high throughput is just increasing the concentration of DNA ends that are available to bind to those nanopores. So right now with the current chemistries, the nanopores just require a lot of DNA as input. It's now possible to recover your library from a flow cell that's spent, and you can put it on a new flow cell, sort of keep iterating that until you have the coverage that you want. So that's quite nice. And with their newest generation of chemistries, they're getting those input requirements down. For ultra long sequencing, you still need, you need even more cells. You probably need 6 million cells to do a proper ultra long run. But for shorter read runs that are more, let's say around 20 kb, with the newest chemistries, you can get away with about 50 nanograms of DNA. So that would be as few as around 10,000 cells.
Great.
So this will continue to improve over time. But
great. All right, thanks. Barak, I wanted to come back to you and ask a question. You talked a lot about the context dependence and of enhancers. It seems like single cell approaches, understanding what's happening in a single cell or a small group of cells that are behaving similarly, would be very important. That you have the potential of losing signal if you're averaging across bulk RNA-seq. How much of a problem is that, given that everything works better for bulk? Single cell approaches are coming along, but how important is it to drive towards single cell approaches for understanding this enhancer promoter interactions in detail?
Yeah, I didn't really touch on it, but extremely important. So really, if I think about what is the underlying magic of biology, it's that every cell in your body has the same genome, roughly, but there are hundreds of different cell types, and now with single cell methods, maybe thousands or even many thousands of different types of cells. And the primary way that you get different cell types is by expressing different batteries of genes. And that means that these gene expression programs are often exquisitely cell type specific. And so, yes, when you average over bulk methods, you get the average. So there are certainly, and I think we're still developing our mental picture of this. I think there are certainly some very general types of transcription factors which are active across multiple cell types, but many of the really interesting ones, especially in relationship to disease, because if you think about it, mutations that affect all cell types, right, generally result in lethality or really severe, really severe types of phenotypes. And so a lot of the disease-causing variants end up being in these very cell type specific regulatory elements. And so in that sense, yes, the single cell methods are really, really important. You are also correct that they are a lot less sensitive. And so I don't know, I don't necessarily, I mean, I assume that the single cell methods are going to continue to get better and better and better because they're sequencing based, and as sequencing gets better, these methods are, will get better. There may be inherent limitations about, when there are a limited number of molecules in a cell and you have an affinity reagent that has to capture it, there are some physical limitations as to how well you can expect to capture those. So in my lab, the way that we do this is we generally will start with bulk and go across cell types to look for the signals. And then once we find the signals that we're interested in, we will go in with single cell methods, sort of more targeted at particular locations. So short answer, yes, really important. We need good, the more these technologies drive towards single cell, the better. They're generally not quite there. And so we're all sort of, I guess the funny thing in the single cell field, really, maybe it's not funny, but the first thing anybody does with their single cell data is aggregate across, multiple cells, because nobody really trusts the profile of a single cell, right? So the question, will single cell methods ever get to the point where you literally trust the profile you're getting out of an individual cell? I don't know the answer to that yet.
Well, and presumably some cells have quite similar functions and clustering is appropriate, but sometimes they don't, and the differences are probably important. All right. Do you guys have questions for each other?
Yeah, I do actually. I got a question for Nick. I was interested in the reach that you described of the immunotethered enzyme, but I didn't quite pick up exactly how far that reach was. Could you comment on that?
Yeah, so most of the methylation is happening within about 250 base pairs or so. And you can, it decays with a half-life of about 170 base pairs. So
okay, so you think it's jumping across nucleosomes as well, because presumably that would go through a nucleosome.
Yes, so we do see at CTCF sites, which, phase nucleosomes around them really tightly. We can see little dampening waves going out from the CTCF site, which would be consistent with linker DNA in between neighboring nucleosomes. If we had to guess, I think we might estimate the reach in physical space to be about maybe 10 to 20 nanometers or so. And presumably we could,
Go ahead.
Go ahead. I was just going to say, we might be able to modulate that by changing linker lengths, right? Yeah.
All right, we're at the top of the hour. Barak, Nick, Bryan, thank you very much for your presentations. Anyone else who is online and didn't get a chance to ask a question, please feel free to email us tdcc@jax.org, jax.org, and appreciate your coming and participating today. Thanks a lot. Bye-bye.